Well, welcome everyone, and welcome to the One Startup World 2023 Summer Conference. It is the premier global startup conference dedicated to helping innovative and disruptive startups succeed. It is the perfect platform for startup founders to pitch their ideas, gain valuable insights, and connect with other like-minded entrepreneurs, industry experts, potential investors, and partners, learning from the best in the business. And now it is my pleasure to introduce Kurt Swogger, the CEO of Molecular Rebar Design, and he will be talking about breaking barriers in biomolecule transport for cells and tissues. So, Kurt, I hand over to you.
Well, thank you, James, and thank you to the team. I really appreciate the chance to talk. So I'm going to start with a little background before we get to the Biopack adventure. Say that I worked for Dow for 37 years. I was lead vice president for thermoplastics, and like everybody else back in the day, decided to make nanomaterials work. Spent a lot of money in our central research group over 6 or 7 years and got what I call a busted flush. Didn't work. And so we passed on that opportunity until I retired. And then literally April Fool's Day 2008, my partner and I got together and decided to make nanomaterials work, specifically carbon nanotubes. I will say there was a fair bit of alcohol involved in that whole discussion, but what we ended up with was actually making it work. So our master company or holding company is Molecular Rebar Design, which is what the label is on this. And our whole idea was to basically invent, patent, develop applications, and then find partners to commercialize things in rubber and batteries, 3D printing, electronics, and so on. And we have an operation just outside of Austin. We have an ISO certified 100-ton plant. And we control and own a majority interest in Biopex Cellular Transport. By the way, the 100-ton plant doesn't sound like much, but we sell this material for $100,000 to $400,000 a ton, which gets all the zeros back, except for the cellular material, which goes for quite a bit more. So what I wanted to show you is what we did to make it work. Is you basically, when you make carbon nanotubes, we were buying the stuff back in the 90s in bundles. And they're 2 to 50 micron bundles, and they're toxic, they're dusty, they're pretty difficult to work with. What we figured out how to do is take one of those 2 to 50 micron bundles, make it into nanotubes that are 15 nanometers in diameter and about 800-900 nanometers long. There's no metals, and we basically formulate them, functionalize them if you want, to be able to make the discrete tubes. And these things are used in rubber. We're going to have Goodyear build tires for us, and the tires are less weight, wear better, and use less energy. Batteries that charge faster and hold more energy, and so on. But one of the things we were concerned about is what are the tox properties, and make sure that we didn't create something that's more toxic. So what we did is, for testing, is we checked, well, if you inject mice with bundles of nanotubes, you get a 0 survivability, that's the red line. If you put our stuff in, the mice don't even notice it, 100% survivability. So we were very excited about that, as you can guess. We then looked at, well, where does the tubes go once they're in the body? And we had organs that take up very little, some and then more. And you notice they all purge out of the body, except for the bone. This actually was a second formulation. We had a formulation where they too purged out of the bone, but we wanted to see if we could get our tubes to stick to the bone for a variety of reasons. So the good news is you put it in and it comes out. Then we looked at what happens if you mix it with a cell, And this is really fascinating because what happens is the tubes actually go into the cell with no damage. The only thing we could figure out or find differently is it did stimulate cell growth a little bit. And we said, gee, that's interesting. But we were mostly concerned, was there any damage to the cell? And then later on, was there any damage to the chromosome? And there wasn't. Finally, we didn't do this work, but we found people who were actually noticing that if you took the tubes, not the bundles, but the individual tubes, functionalized them and so on, the body actually basically destroyed them over time. And they actually looked at a wide variety of tubes. The ones we happen to like, the half-life's 41 days. So we don't put much in. And if you do, it either comes out in your urine or feces, and anything that's left is basically digested. Now, for our commercial applications, this is wonderful. And we were very excited because now we knew we could handle these materials much easier, and we won't get into that, but there's no issues with toxicity for people working with them. But when we look at what did we have from the nanotubes, they're non-toxic, they're very simple. We don't have any size limitations on what we can absorb, whether it's a really big protein or very small chemotherapy. These tubes are very reproducible, very scalable. We can milk them by the ton. They're very cost efficient. They tend to protect what's ever on the surface, and we can target them. And so what can you do with this kind of material in the body? So one of the things we find out, and by the way, we do all this testing with outside labs. Charles River is the group we typically use. We found out we could take and deliver MGMR, could deliver chemotherapy or other treatments to tissues such as bone and liver, the easy ones, and then we could make it work for other organs. And one of the interesting things, we showed that doxorubicin could go and cure cancers, particularly bone cancers, and have zero side effects that anybody could detect. So the other thing we could do, we only had to put a third of the amount of doxorubicin in, and we could go to 2x. So the doctor had a lot of range in what he could treat the patient with. So at this time, we're looking for a partner to accelerate this idea of chemotherapy or other drug with no side effects. The second thing we looked at is we can deliver anything particularly we want into that cell. So we actually have a project with the Army, and we're delivering plasmids into cells to generate antibodies to attack HIV. And we're actually in a $1 million phase. We're working with Texas A&M, who will be testing this in mice. And if that works, we'll be following it and testing it in primates. And being able to see if we can knock out this particular drug. Much to our amazement, we had no idea that $25–30 billion are spent for HIV therapy, jumping to 40 by 2030. Finally, we also can take and enhance cell growth, and we can do that for individuals. When you're going to lab and having your, say, stem cells, people want to replicate, and we can go up to 60 to 80% faster. We could also do it in big vats of chemicals. And so this one, we're working with UT San Antonio on a couple applications. And besides stem cells, we're pretty sure we can do wound or burn care applications. So if we look at what's going on out there in the real world, this is the materials, viral vectors, polymers. So this is what people are using for this cellular technology. And when we look at what we do versus them, we have a much broader range of application. Our tubes don't care about its cargo. They don't care really about it, and they don't have toxic properties. We can make lots of it if we need to. They also enhance stability, very minimal processing steps compared to the other players. So this is actually the one we're working on right now, and we've made this work. Basically, we take our MR, put it on a plasmid, deliver it into a cell. The cell then, the plasmid desorbs off the tube, goes in, changes the chromosome and DNA, begins to make antibodies. And the stage we're at right now is we've transferred this to A&M where they're doing it in mice. We're not even worried about the toxicity of the tubes for mice because we've done hundreds of them. But what we're really interested in, how much of this antibody will we make? And the good news is the target's muscle cells, and there's lots of them. So when we do this, it's worked in the Petri dish, if you want. We'll go into mice, and then if that works, like I said, we'll put it in monkeys. And in case you want to know, no, the monkeys aren't destroyed. They're actually repurposed. So where we're at is we've been working on this for a while. Again, we're looking for, particularly in HIV, a partner. The third phase of our defense contract would be find a partner to make the product. We also are looking for a chemotherapy partner, and I'll say the stem cell materials, we're still doing research, but that also there's a place for a partner. And I can't talk about the application because we're under an NDA, but that one, we need a partner for it pretty nicely. And it's a really big market, is all I'll say. So we actually have a pretty good revenue stream coming into the base company, not particularly BCT. We have a lot of published study, 140 patents covering this thing, all compositions, lots of collaborations to make it happen. And what we're looking for is partnerships as much as anything else. Who can we partner up with? Somebody that wants to develop a lower cost cancer treatment, which would be the chemotherapy, or cellular transport, HIV or other diseases. We also would like investment. We talk about that if somebody's interested, and that's to develop a GMP facility. We've got an ISO 9001 facility, which isn't quite what we need. We want an FDA master file, and we want to continue to develop more applications. So we'd love any advice or feedback and discussions. This is the neat thing about this, it's a chemical engineering's dream, because you're really doing mass transport of really neat chemistry that can help the body. So with that, I'd like to say thank you so much for the time, and I'll be glad to take any questions. They did ask me if I wanted to do demonstrations, and the only thing I could think of was bring in a bunch of mice. And I knew then I'd lose them and my wife would kill me. So we just have to skip the demonstration. Sorry.
Well, thank you. Thank you very much, Kurt. This is an extremely interesting presentation. And it looks like you get rid of all sources of intoxication from whatever materials are used to inject or introduce subjects within the body.
Well, I'm not so sure we can take out alcohol, but we do a lot of other stuff for sure. We're really a transport vehicle.
Yeah.
And if you want the biochemist to have a really fancy chemistry, their problem is getting it where they want it to be.
Right.
Not where they don't want it to be.
Right.
On any of these applications, we can surely say, well, why do you get chemotherapy with no side effects? All has to do with you never expose muscle and nerve to the agents. You just go where you want it to be, and the other stuff's purged very quickly.
Yeah. Yeah, I understand. You have patents for these things?
Yes, sir. We have 140 of them. The way we work it in the medical field, there are access to all of them. And there's maybe 10 or 15 very specific to the areas we talked about.
Right, right. And are you in any way looking for partners?
Absolutely. We need a partner. We'd love somebody that wants to build new formulations for chemotherapy. You literally could take doxorubicin and reformulate it, and it would work just fine. There's no side effects. big problem, though. It's be much cheaper than what's out there today.
Right.
People are not very excited about that. But outside the US, people can't afford $70,000 a patient. Inside the US, I guess they can.
Yeah. Yeah. And are there any external factors that are influencing what you're doing, in some way interfering or creating a blockage or something like that, or is the way is open?
The way is open. I think what we need to do, we kept very quiet for years to get all these patents. And so we're not well known. So when y'all called, I said, well, yeah, we'll expose, see if there's some people out there interested in this. And but you know, the same goes for, nobody's heard of molecular rebar unless you're in the rubber business or the battery business.
Right.
And the same thing goes here. And we're fine with that, but now it's hurting. It helped us get the patents. And so the patents we have are all composition of matter and application patents.
Right.
You can't find how we actually do what we do anyplace, but
Ah,
all patented.
well, it sounds terrific, and we're very much at your disposal to see if you can find good partners, and any way we can help, we'd be happy to do that. This seems like a very innovative idea. I can't claim to understand all the details and science behind it, but it makes good sense, and we wish you all the best, and we will be here to help you as much as we can.
Yep, some folks that want to do partnerships, and particularly if they want to invest in the company. It's a C Corp. We did do a crowdfunding round, so we're all set to, if somebody wanted to invest, happy to have them.
Terrific.
All right, well, thank you so much.
And thank you, and we look forward to staying in touch.
I know, thank you. Bye-bye.
Bye-bye.